Today, we're going microscopic. We're going to follow one of the most incredible search and destroy missions in all of biology, and it's all happening thanks to a revolutionary tool called CRISPR.
So first, let's just get our heads around the sheer scale of this challenge. The human genome, right, has billions of DNA letters. Finding one specific gene sequence and all of that, it's like trying to find a single unique book in a library the size of a city. And oh, by the way, all the books are written in a 4-letter alphabet. It's a truly monumental task. And if you could shrink down to that molecular level, this is kind of what you'd see. It's not some neatly organized bookshelf. It's a crowded, tangled, chaotic world of genetic code. So how on earth does our gene editing agent navigate this mess to find its one precise target? To pull off this mission, science has basically recruited an incredible molecular agent, a protein that's been perfected by nature over millions and millions of years. So let's meet it. The technology is called CRISPR-Cas9. And here's the really cool part. We didn't invent it from scratch in some lab. We actually discovered it in bacteria. For them, it's a super sophisticated immune system that they use to fight off invading viruses. They literally chop up viral DNA and store a piece of it, like a most wanted poster, so they can recognize and destroy it next time. We've basically hijacked this ancient defense mechanism for our own purposes. So the whole system is this brilliant two-part marvel. First, you have the agent itself, the Cas9 enzyme. This is the protein that does the heavy lifting, the actual cutting of the DNA. But here's the catch. It's completely blind without its mission briefing. And that briefing comes in the form of something called guide RNA. The guide RNA is a little piece of genetic material that we can program with the exact address, the GPS coordinates, of the gene we want to find. All right, so with its coordinates locked and loaded into that guide RNA, the Cas9 enzyme is let loose inside the cell. But how in the world does it find its target so fast in that crowded library we just saw? Well, it doesn't just drift around hoping to get lucky, randomly bumping into the right spot. Nope. Cas9 is an active search agent. It uses this really clever process to scan the genome with incredible speed and efficiency. It's not passive at all, it's on the hunt. And this shows its two main strategies perfectly. For short-range searching, Cas9 will actually latch onto the DNA and slide along the strand, kind of like a bead on a string. But to cover huge distances, it hops. It makes these little three-dimensional jumps through the nucleus to land on a completely different part of the DNA. It's this combination of sliding and hopping that makes the search unbelievably fast. And now for the climax of the whole mission. That moment Cas9 actually finds its target and locks on. This right here is where the real magic happens. You might think that Cas9 is constantly checking every single DNA sequence it passes against its guide RNA, but that would just be way too slow. Instead, it has a shortcut. It first scans for a tiny, specific landmark that basically screams, "Hey, you're in the right neighborhood. Now it's time to take a closer look." And this landmark has a name. It's called a PAM sequence. For the most common type of Cas9, that sequence is just 3 letters, NGG, where that N can be any DNA base. Think of the PAM as a universal keyhole. Cas9 is just zipping along the genome, and only when it finds this specific keyhole does it even bother to stop and try its key, which is of course the guide RNA. The second it finds that PAM, an absolutely incredible physical transformation begins. If you are watching a high-res 3D animation of this, this is where your jaw would hit the floor. The Cas9 protein isn't some static, rigid tool. It's a dynamic molecular machine. It literally changes its shape, with different parts shifting and locking into place. It reconfigures itself in real time to grab onto the DNA. So it's a multi-step verification process, which is just genius. First, it docks at the PAM, check. Then, using its own power, it forces open the incredibly stable DNA double helix. It literally unwinds it. And then, and only then, it brings in its guide RNA to see if it zips up perfectly with one of those now exposed DNA strands. If the match is perfect, the mission is a go. Okay, so the target has been confirmed, everything's locked in place. Cas9 now completes its primary mission, making a precise, clean cut in the DNA. And this is not a clumsy chop. The Cas9 enzyme has 2 specialized cutting parts, these domains called HNH and RUVC. They work together like a pair of hyper-precise molecular scissors. One domain cuts one strand of the DNA helix, and the other cuts the opposite strand, creating a clean,
double-strand break exactly where it was told to.
So after Cas9 makes the cut and clears out, the cell's own emergency repair crew rushes to the scene, and it has two main ways to fix the break. The first way, NHEJ, is kind of the quick and dirty approach. It just shoves the two ends back together, which often creates little errors that can shut a gene down. But the second way, HDR, is way more precise, and this is where scientists can step in. We can provide a correct DNA template, and the cell will use it to repair the break perfectly. This lets us not just delete a gene, but actually rewrite it.
So just think about that. This entire molecular mission, this search and destroy strategy that was perfected in tiny bacteria, has now become one of the most powerful and revolutionary tools in all of modern science. I mean, the possibilities are just staggering. We're talking about using this to disable the genes that cause devastating diseases, to correct genetic mutations that people are born with, or even to insert new beneficial genes. We can even modify Cas9 so it doesn't cut at all, but just sits on a gene to turn it on or off. It has unlocked a completely new frontier in biology and medicine. You know, the power to find and change the very code of life with this kind of precision is just immense. The question really moves from can we to should we? And that leaves us with one of the most important and complex conversations of our time.